So now we're talking about membranes. It's really kind of vital to talk about their permeability. Okay, if this is like a castle wall, that's great. That wall has to let things through, right? Things have to come in, things have to go out, right? You can't just be under siege forever, right? Cells would die if that was the case. So, what is it? How do we keep the enemies out and let the good stuff through? Well, there's a whole variety of things. If you look at lipid soluble molecules, like fats, for example, right? They have a much easier time getting through. And the main reason for that? Well, they're already lipids. If there's a famous rule in chemistry that like dissolves like or will incorporate like. For example, if you get a red wine stain on a carpet, one of the best ways to get rid of it is with white wine, because the white wine can help solubilize the red wine, and you can get it out of the carpet. But I don't recommend dropping any form of wine on your carpet, especially red. Ooh, couldn't imagine white carpets. But membranes themselves, they're naturally permeable to lipids, right? And some viruses can be pretty nasty that way, where they'll try to sneak in because some viruses have stolen a bit of membrane on their way out of their previous host. But lipid soluble molecules tend to pass through a bit more easily. But like big ones, like large proteins or charged molecules, that's a little harder to get through because you might interface at the phosphate heads, but then you have to get all the way through that hydrophobic region, right? Where the tails are. And that's a little bit more of a game. So what do we have to do now? Now we have to look at just how transport works. So there's really three types of transport that I'll go over. Only one requires energy. So let's nail that right away. This one, and I want a better color than yellow because that's not working out for me. There we go. This is the only one that requires energy, active transport. And it's got a great name because think of it, when you're active, you're expending a lot of energy. So that's an easy one to remember. Okay? This is the only one that you're going to need energy for. Okay? The other ones, well, they don't require energy, but they can still transport items across the cell membrane. So it's probably piquing your curiosity. Think about it this way. When you're experiencing a lot of thirst, when you consume water, water doesn't have to be pumped across the cell membrane. In fact, water is really tiny. And if you remember what I was telling you about water, it can get through pretty easily. And remember that when we have this sort of castle wall, we have proteins, we have little doors, if you will, things that allow things across of a certain size. We also have certain proteins that will just allow things through in an easy fashion. They don't even require energy. But I digress. We should see that in action. So what do we have to do here? Well, we'll go back to diffusion, and then I'm going to show you some animations in a minute. Diffusion is the easiest one. Diffusion, please don't say water when you say diffusion. Okay, no water here. Okay, diffusion is when something passes from a high concentration to a low. So here's a great example in school. When somebody sprays Axe, which drives us all crazy, it's strongest where they spray it, and then as it spreads out over time, it spreads out into its low concentration environment. It just spreads out. Pretty easy, right? Diffusion is when we're talking about things other than water, okay? And when I talk about diffusion, I will do everything I can not to talk about water, because when we talk about water spreading out, that's different. We're talking about water spreading, like right now, I'm quite thirsty. If I have a glass of water, that water will spread from its high concentration region, which would be my digestive system, into the low concentration region, which would be my circulatory system, which really wants a drink. Just carry the water molecules throughout my body and to hydrate my body. So please, 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 I'm being so nice about it. Diffusion and osmosis. Not the same. Okay. It's odd. I need a different color. There we go. Okay. They're both transfer from high to low concentration. That's true. But diffusion is when we're talking about water. And, big one, that water has to go through a membrane. See, we can talk about diffusion of Axe spray in the hallways. We can talk about diffusion of food particles in the gut getting into the bloodstream. Sure, that's diffusion. But when we talk about osmosis, it's water and membranes, folks, and don't get it wrong, okay? Now that I'm done ranting about that, there's one more that I want you to be aware of, and it's a weird form of diffusion. It's a kind of diffusion that's sort of helped out. Now, you can have these little proteins in the cell membrane that help out diffusion. They don't require any energy. Sometimes these proteins, when they encounter something, will sort of change shape based on what they touch, and they act as transporters. A really good example of this is when you go to the pool, and I'll draw it, and the water flows into one of those little buckets that looks like this. And when the water fills up, the bucket turns upside down, right? And it doesn't take any energy, right? Such a great drawing. There goes all my water. Right? When this bucket turns upside down, the water is just released down to all of us having a great time. If you've been to Great Wolf Lodge, there's a huge bucket in this water park that does just this thing. And every time I look at it, I think about facilitated diffusion, because the bucket itself is like one of those proteins, and as it fills up, when it reaches max capacity, it tips over and the water flows out. But that's not any energy. The protein itself isn't running like a generator or anything. It just flips. And that's one of the many ones we'll look at. But for now, facilitated diffusion is like diffusion with protein help. Yeah, I like that. That's actually kind of deep. Active transport is the one that's going to require energy. Now, in this course, when I say energy, you say ATP. Adenosine triphosphate, folks. Okay? That's the energy molecule that our bodies actually run on. We all think that we run on glucose and that carbohydrates are really all of it, hydrocarbons, but that's not the whole story. We need all that stuff is important, but it's used to make the fuel, which is adenosine triphosphate. Think about it this way. We might run a generator to power up our house or to power up our cabin, but the real power is the electricity, right? That's what actually runs everything. Yeah, so what actually runs things in our body is ATP. Okay, now I'm going to sneak out of this and talk about, there we go, diffusion and all that great stuff. So I had an animation here, great little animation, and what we're looking at is the bilayer. Right? There's our fatty acid tails, and got our proteins, some going through, some not. But what's really important here is to look at osmosis and diffusion. Now, for those of you who've done a lot of chemistry, what we're really looking at here is an equilibrium. So think about it. If you eat a meal and your let's say your blood transports those nutrients throughout the body, we want to get those nutrients from the high concentration outside the cell into the cell. That's the whole point. So if you think about the nutrients passing through, they're on a high concentration on the outside, right? There is a net flow, right, towards the inside of the cell, because that's just how diffusion works. If you drop, just put one drop of food coloring in a glass, it starts out really concentrated where the drop falls, but then come back an hour later and it's spread out. And if you used red food coloring, you've got sort of a light pink to the half glass of water, for example, that you might have used, right? The point is, this is, when we look at diffusion, it's an equilibrium. because there's so much more stockpiled outside the cell, there will be a net sort of flow to the inside of the cell. Now, there's always a counterflow, but it won't be as high because this is called a gradient, a concentration gradient, where the concentration gradient is greater to the inside of the cell than the outside of the cell. So if you've got a lot of Burger King out here, it wants in. Okay, I like Burger King. True story. So when we look at these kind of processes, if the molecules aren't too big, for example, they can go straight across. Now, if, for example, we'll say lipids, little mayonnaise particles here, they can get across quite easily. That doesn't require energy. So what we're looking at is something called passive transport. Diffusion and osmosis are both passive transport. So here's a little animation, and it shows the particles flowing to the inside of the cell. Now you'll notice that we're at an equal concentration right here, okay? And this is called an equilibrium. And that's what really the point is of diffusion and osmosis. Now, this is a neat little experiment. If we take two balloons and we make this one really, really, really high in sugar, we make it super sugary, and this one lower in sugar concentration, you have to wonder, well, put a membrane in between the two. It has to be like our cell membrane. It has to be semi-permeable, right? What's going to be the net flow? Like, we've got high sugar concentration there, low over there. What would happen? Ponder and ask yourself that question. What's the net flow? Which way would it go? Well, let's see. Now, this is a flow of water. Now, this is interesting. These are sugary solutions, but this one on this side was so syrupy and yummy that the water in this side was drawn over here. Think about it this way. When you get really, really thirsty, right, what your body's trying to do is it's trying to water down all that concentrated material in your blood. So this balloon right here, its water rushed over here, can get through the membrane really easily to try to dilute this down. Remember, water particles are quite attracted to molecules like sugar and salt, things like that. So they'll just flow right across. And in these kind of experiments, we do these in class where you can say, well, this was actually the more concentrated region, so the net flow of water was in this direction. Hey folks, that doesn't require energy. That's what we call a net osmotic flow. Right now I'm really thirsty. So what will happen is water will flow out of my digestive system and into my bloodstream just like this to try to water down what's going on there. Some pretty neat stuff. But folks, it's kind of all about passive transport. And when we talk about passive transport, we kind of need to look at some of the transporters. So I've got some neat stuff here for you. Now, in some cases, this is a glucose transporter molecule here, and if you notice, this protein in our cell membrane has the same conformation as the glucose molecule. Remember, glucose is a hexose sugar, right? Six-sided sugar, and this is a very neat piece of evolutionary engineering because when the glucose binds, this is kind of like that that water container I was talking about, telling you about at Great Wolf Lodge. What will happen is once it fills, the protein sort of undergoes a little change, kind of like the bucket, and releases glucose to the inside. Okay, now this is a very neat trick because it gets glucose into our cells. Okay, there was no energy involved there, right? It was just a change in the structure of the protein. So that is a passive mechanism. Okay, and that's a neat little. That's actually an enzyme called glucose permease. Neat term. Glucose enzymes end in ASE, right? And it's the enzyme that allows glucose to get through the permeable membrane. There we go. Great fun. Now, if you look at this en masse, you can see what's happening here, right? The glucose transporter what happens is you kind of can get an equilibrium. So it's not like it stockpiles glucose. We're not. It's not like running a machine to get it in high concentrations, right? But it does get glucose into our cells, and that's good. We kind of need it, right? It's great fuel. Okay, so I think I've shown you this one already. Now, active transport is a little bit different. Active transport is when we pump things across. Okay, so in the case of active transport, for example, our nerve cells are like this. We use a little bit of energy to pull things in and send things out. Now, this little trick here, you can see right there, that forced some sodium out and some potassium in. And this is, we'll talk more about this when we talk about your nerve cells. This is called a sodium potassium pump. But what it does is it uses a little bit of ATP right there to get particles where we want them to go. And if we're going to pump something, it's true, you're going to have to expend energy to do it. Nerve cells won't work unless you see this play out in sort of this fashion. Okay, so I can get rid of the stabbing balloon trick. This is another neat little one. I'm just going to, I have to mute it because it's so loud. But just to review passive transport, there's the extracellular fluid, intracellular fluid, right? Here's all the little parts that we all know about. And in this case, this one looks at oxygen. So oxygen being a high concentration in our blood needs to get into our cells. The net concentration gradient, there's more oxygen on the outside than on the inside. So it's coming in. That's diffusion, folks. And here we go. That's really just how it goes. This is what our red blood cells do to deliver oxygen. And you notice it didn't have to go through any of the protein structures. It didn't need assistance. It's very small. Osmosis, one more view. All right, there we go. So water itself is charged, but remember, it's a very small molecule. So if right now I'm feeling quite thirsty, high concentration of water after I have a big old 1 liter of water, it's going to come through because that is the gradient. I'll give it a moment there. And it comes right through. Voila. Now, facilitated diffusion, okay, so there's a little bit of protein help to let something through. So again, there's our players. And in this case, the protein itself is going to look like a pore. Some of these proteins are porous to allow certain things through. So here's another way to get glucose in. Check this out. So if we had high concentration of glucose on the outside and low because our cells use it up as an energy source, well, the net, based on what we eat, the natural concentration gradient is going to be inward. So let's see how this glucose trick works. Nope, can't get in through there, but maybe, just maybe, it can sneak in through a porous integral protein like this. Absolutely. Okay, and that facilitated the diffusion to happen. Because glucose, think about it this way, this glucose isn't going to be able to just sneak through the polar heads. This is a large molecule, okay? I know the size of these may be a little misleading the way they're being drawn, but it's a very big molecule. So it needs some help to get in. And that's the role of some of these proteins. This is the flipper model, just sort of seen one more time, right? Where there is a change in the shape or conformational change, and glucose binds to it without consuming energy. You could see that the gradient, glucose wants to get in because there's not as much on the inside, and flip. Pretty neat. So glucose flipper. There we go. And then active transport. This animation doesn't show active transport the kind of the way that I wanted to, but it is neat to look at some of these things. Just going back to this, I promise this is the last one I'll show you here. Osmosis. When you look at osmosis, and you see this is water, and here's that semi-permeable membrane, you see that it's in equilibrium here right now. See that? Equal amounts on either side, right? They're just, these water molecules are bouncing around, practicing Brownian motion, right? Just motion because of the natural energy in the system. But if you add something like a salt, see this level go up? Well, remember, this is charged. So oxygen, which is the polar end of the molecule, loves this. And chlorine, which is a negative ion, while the hydrogen ears, which are positive, love hanging out with chlorine. So what you'll see is these are exciting. And because these are charged particles, these water molecules, which are polar, will come over and sort of play ring around the rosy around these things and surround them. And there's a net osmotic flow in this direction. This is what happens when you eat chips, folks. This is why you feel thirsty. This is why your blood pressure goes up, because more water flows into your circulatory system because of all these little particles. It's the exact same thing when you consume sugary snacks, right? It's bad, bad, bad for your system. Okay, I think I could sneak back out and finish off a little bit of a little bit more material. My iPad's long gone to sleep. Okay. And I have to make sure. This is great fun. Right there. I have to turn the mirroring back on. There we go. So getting back to, there we go. Getting back to just sort of our slideshow notes, diffusion doesn't require any energy. No energy. Awesome. So the concentration will spread out. A little graph down there used to be animated, but the movement is always from the most concentrated region out. Again, just sort of lipid-soluble molecules. Alcohol, actually, one of the things, there's a little rule about alcohol: beer before liquor, never sicker. What it means is beer itself is a kind of a molecule that is in your stomach, will just go right through the lining of the stomach, and it'll make it into your bloodstream very quickly. The reason they say beer before liquor, never sicker, is because the carbonation actually accelerates that a bit. And to consume harder alcohols like whiskey and things like that after consuming beer, you've already increased your permeability rate, and now there's much more sort of nasty alcohols going into your system, and your body can't keep up with processing it. But lipid soluble molecules, alcohol, these gases, oxygen, carbon dioxide, things like that, they go through the membrane very easily, right? And thus the comparison I was using to say they can get into your blood really fast, right? Not good. Water can pass through channel proteins, but it's actually pretty small, so it can get through as well. It's not a very large molecule. But big things, right? Like, for example, glucose, we were talking about. It's when we're talking about large molecules, right? Glucose, for example, I'll just draw its six-sided ring here. Proteins, amino acids, they're pretty big. They're hard to get through. And charged ions, they have a hard time getting through. They need little sort of doorways, think of it that way, in the castle wall to get through. So they're going to need help, whether it's a facilitated kind of help, like what we saw with the glucose transporter, or whether or not it's a pump and it's active transport. The thing about active transport, and I want you to think about that, is active transport is a way to stockpile things inside the cell. If you want to get lots and lots and lots and lots of stuff inside the cell, you can pump it in. And that, you're sort of defying the laws of diffusion and osmosis, because what you're doing is you're stockpiling. And that you would think that stuff would get out. But if you pump glucose in like mad using energy, it can't get out. And that's why we have active transport. We want to get all that those nutrients stockpiled inside the cell so that it can continue to live and thrive and divide. So osmosis is it's a very interesting thing to sort of show people how one side of a container can rise because it's more sugary or more salty, as in the example I showed you. When you look at your blood cells, you have to be aware of what that could actually mean. There was a case of a lady that had drank so much distilled water that it actually killed her. And you've got to watch out, because if you consume too much water, your cells themselves are considered much more concentrated than the watery environment, and water will just flow in and flow in and flow in, and that can cause cells to burst. It's not good to drink distilled water, folks. Okay, it just isn't. You can actually kill yourself doing that. But on the opposite side, if somebody gets so thirsty that they can't get enough water, what will happen is water will just flow out to the point where your cells, the membranes, start to collapse on each other, and they'll look almost like these little spiky balls, and then they'll just shrink, shrivel, and then they can't do their job anymore. So again, I go back to homeostasis, saying there is a fine balance. Okay, very, very fine balance. But osmosis, if you want to be completely technical about it, is the diffusion of water across a semi-permeable membrane. Okay? But it's better not to use the term diffusion, because you'll start to use it too liberally. So what you want to say is osmosis is the net flow of water across a semi-permeable membrane, or barrier, as this slide suggests. So in this case, you can see the water molecules are here, right? Water molecules are here, and what they'll want to do is, the membrane is this region right here, and you have to say, well, what is the net flow? Well, if you look at this is the inside of the cell, right? Because it says cytoplasm, and this is the outside right here. Oop, change colors. What will happen is there's more water particles on the outside, right, than on the inside. So your net osmotic flow is in this direction. And folks, we can actually measure that, because we can see, we do this in the lab, we can see how much water levels rise by depending on the concentration of different materials. We can make things sugary or salty, and we could say, well, how much water flowed in? We could say, well, this many millimeters flowed in over this time. We could actually set we can attach a set of units to it, right? You could have milliliters per second, milliliters per minute, milliliters per hour. So net osmotic flows are things that can be measured, which is very scientific. Yay! Okay, and I'm going to stop here. Hoorah! Because this leads me into a conversation about how strong solutions can be. Are they weak? Are they sort of middle of the road? Or are they super concentrated? And this is a totally different lesson. But for now, I'm going to stop. So I wanted to thank you for putting up with me, my animations, and all my crazy stuff. And we'll see you another time for eCampus Biology Live. Thanks, folks. Ciao.